our last topic and may reporting. Okay? Para lahat naman kayo magsalita, hindi na ako yung dada ng dada dito. No? Ah, i Okay, yung mga queries niyo sa quiz mamaya na lang. No? Saan na tayo? Yung topics for reporting sasabihin ko na lang sa inyo mamaya, pero that's applied bacteriology. Okay, for food, water, industrial bacteriology. Groups will be sent this week or baka mamaya isa-send ko na rin para masimulan. Okay, so can we start? Pwede na, no? Okay. Okay, so good morning. For this morning, we will talk about the non-enteric gram-negative bacilli. And bago pa lang lahat, I expect you na sana napanood nyo na yung mga sinesend kong links and PowerPoints modules. Kasi yun lang yung way na alam ko para may deliver ko din yung topic. Kaya mag-alala, kami din sinesendan ng ganun at yung reaction ko, may typical reaction is, naasar ako kasi bakasyon din nagsisend ng link ng lecture video. So maybe some of you naasar, pero yun lang talaga yung paraan para ma-deliver yung topic sa inyo. Okay? So as a student, lahat naman tayo student, comply na lang tayo. Diba, bakasyon naman na next month, malapit na. Okay. So for your non-fermentative negative bacilli, first we will discuss their general characteristics. So they're actually the opportunistic pathogens in the human, in us, sa mga tao. They are found in food, sa environment, and especially sa mga hospitals. No, we will also discuss the OF test, which I believe na ilang beses na rin natin itong nabanggit, na the oxidative fermentative test. The Pseudomonas, okay, si Pseudomonas, marami itong karakteristik na dapat itackle. Yung mode of acquisition, the clinical significance. Ito rin kasi yung bacteria na mostly pigmented and they have their distinguishing characteristics. And the other species of Pseudomonas and the laboratory diagnosis. Okay? The Stenotrophomonas maltophilia, Burkholderia, and Acinetobacter, their biochemical reactions and the different species under these genuses. And lastly, the other non-fermentative gram-negative bacilli. Okay, wait lang. Before taking your quizzes, pakicheck naman kung hanggang anong oras yung availability. No, so mga quizzes, yung queries nyo sa quizzes, mamaya na lang. Okay, PM na lang. So as we said a while ago, your non-fermentative gram-negative bacilli, they are opportunistic pathogens commonly found in water, soil, plants, vegetables, food, and even in hospital surfaces. Ang common dito si Pseudomonas aeruginosa, no? Especially sa mga NICU, sa mga NICUs, neonatal intensive care units. Lagi silang nakikita sa surfaces. Wherein nakakatakot kasi yung mga bata, yung mga babies, yung mga neonates, they have very low immunity sa mga bacteria na kapag napasok sila ng bacteria, they will have sepsis. Pag napabayaan, will eventually lead to death. Okay, they can withstand the treatments with chlorhexidine and quaternary ammonium compounds. Your quats, okay, we discussed that yung mga quats nyo naman na nung first term, the detergents and your phenolics, right? They can be isolated from nebulizers, dialysate fluids, salines, catheters, and other hospital devices. The most species are they secrete oxidase, and metabolism is primarily respiratory and never fermentative. So they are oxygen-based or oxidative. Okay, ferment carbohydrates, ito, by oxidative method and not by enzymatic reaction and produce a very weak end products. So the acid production is absent in oxidative fermentative media if it is overlaid with mineral oil in promoting anaerobiosis. Okay, ang OF test kasi is ganito. Using the Hugh-Lifeson medium or your OF medium. Okay? So, OF test, this differentiates Enterobacteriaceae from Pseudomonas. So, ganito yung reaction. Ito yung mineral oil, yung black na nakalagay dyan. So, inilalagay yan to promote anaerobiosis, to promote anaerobic environment. Okay, ang kaiba sa Enterobacteriaceae and Pseudomonas, si Pseudomonas, hindi niya kayang i-ferment ang sugar in an aerobic environment. So, ito, anaerobic environment. Si Enterobacteriaceae, they can ferment sugar in aerobic and anaerobic environment. So, yun ang kaiba niya. Okay, oxidative, requiring oxygen. Kaya oxidative. So, Pseudomonas, they are oxidative and not fermentative. Okay, fermentative kasi sa anaerobic environment siya. Diba? So, O negative, O positive, F negative, and Enterobacteriaceae, O and F positive. So, this determines the ability of the organism to utilize the substrate, thereby producing acid byproducts in the presence or absence of oxygen. Ayun, oxygen barrier, mineral oil. In some reference, ang naglalagay sila ng parafilm, which can also promote an aerobic environment. pH indicators are the following. We already discussed this. Okay? May iba-iba silang color in producing their positive result. Okay? Pag open tube for your Pseudomonas, they exhibit a yellow color. Kapag closed tube or kapag in an aerobic environment, in the absence of oxygen, green or blue-green color ang closed tube. Okay? So, they are not fermentative. The Hugh and Lifeson medium is an OF medium with 1% carbohydrate in bromothymol blue as the pH indicator. Okay, the Pseudomonas or Pseudomonads, these are the organisms that are most commonly isolated non-fermentative bacilli. So sila yung most commonly. Yung second is si Acinetobacter. And yung third ay si Stenotrophomonas. Stenotrophomonas. Madidiscuss din naman mamaya. So they are obligately aerobic, non-spore-forming, and motile bacteria with polar flagella. They grow on primarily plate media like your MAC, conky, and blood agar plates and can be found in your cosmetics, in swimming pools, causing swimmer's itch, a swimmer's ear, in hot tubs, in inner parts of shoes. So the Pseudomonas fluorescent groups that fluoresces, they are the Pseudomonas aeruginosa, fluorescens putida veronii, montelii, and mosellui, luila, ay yan. Pseudomonas non-fluorescent groups is Tutseri and pseudoalkalichins. Okay, in the biochemical test, since most of them produce oxidase, so they are positive in oxidase and catalase, and non-lactose fermenter. So in TSI, A over K, they don't produce gas and don't produce a hydrogen sulfide. The agent of blue pus or Pseudomonas aeruginosa is the most commonly isolated species of the genus in the clinical specimen, and this is the most commonly encountered gram-negative bacterium that is not a member of Enterobacteriaceae. So they are monotrichous, okay, rods. Okay, pag sinabing monotrichous, flagella at one pole of the bacterial cell. Okay, when grown in blood agar plate, they have high spreading growth and are flat. Okay, have metallic sheen, parang ganito siya. Blue-green color and some will present a red-brown color, have grape-like or corn tortilla odor. Okay, sometimes mucoid observed with patients with cystic fibrosis and have beta hemolytic pattern. Okay, the virulence factors are endotoxin, pili, alginate, capsule, and the following. And you can acquire this infection through ingestion of contaminated food or water, and from contact lenses, also found in hot tubs and whirlpools. Okay, clinical significance, one of the common causes of gram-negative bacillary bacteremia, causative agent of ecthyma gangrenosum. So, ganito ang ecthyma gangrenosum. This ecthyma gangrenosum, so as you can see here, This is a cutaneous infection and usually occurs sa mga patients who are ill and syempre immunocompromised. Yun naman ang primary reason kung bakit tayo na-infect. Somehow, yung immune system natin bumaba. Or is, ano yun? Basta bumaba. So, the characteristic of the lesion, kung titignan nyo, para siyang ma-red, so hemorrhagic or bloody, bloody pustules that will in turn be evolve to be necrotic ulcers. Okay? So, forming black colors. Okay, so parang ganito siya. So, ganito, mag-evolve siya ng ganito. Si swimmer ears naman, swimmer's ear, so, ganito ang itsura niya kapag infected ka. Ano sabi natin ulit? This is also known as otitis externa. So, typically, caused by water that has stayed for too long sa outer canal ng inyong ear, okay, for a long period, providing a moist environment sa bacteria so that it can grow here sa tinga mo. Okay, so anyone, kahit sino, immunocompromised or not ka man, can get swimmer's ear but is often seen sa mga bata. Okay, so parang bata to, no? And jacuzzi syndrome or the necrotizing skin rash or folliculitis or hot tub folliculitis. So ito kasi ini-infect niya yung roots ng inyong lower parts of the hair follicles. So ini-infect niya yun, kaya ang rash niya is distributed. Okay, commonly seen nakikita sa roots of your hair follicles. So ganyan itsura niya. So etiologic agent of lung infection is cystic fibrosis patient and causes hospital-acquired infections that are related to antibiotic resistance. Okay, si Pseudomonas, they have different pigments. Your pyoverden that produces a yellow-green or yellow-brown pigments. So this is the pyoverden, verdin or verde. Okay, green. Okay, green. And pyocyanin, blue pigments that is only produced by Pseudomonas aeruginosa. So dito, na-isolate, okay, pyoverdin and pyocyanin. Pyorubrin, red pigment, and pyomelanin, brown or black pigments. So makulay si Pseudomonas. Distinguishing characteristics are the following. Okay, positive siya, mag-grow siya in high temperature. Okay, can utilize acetamide as carbon source and can utilize citrate as carbon source. Okay, positive in gluconate production and arginine dehydrolase. Next is your Pseudomonas efflorescens and the Pseudomonas putida. They are isolated from contaminated blood products, cosmetics, even in hospital equipment, pero most commonly si Pseudomonas aeruginosa. Okay, urine and respiratory specimens can grow at 4 degrees and have been linked to transfusion-associated septicemia in transfusing your blood. Okay, differential test, gelatin hydrolysis for Pseudomonas fluorescens is positive. Okay, in gram staining, they demonstrate a straight and slender gram-negative rods. Okay. Hirap. May ubo. Okay, you can grow them in blood agar plate, chocolate agar, McConkey agar, and Seller's medium that promotes pigment production, the same with Cetrimite agar, and in C390 medium. Okay, Cetrimite agar, differential and selective medium that also enhances the pigment production. The Pseudomonas fluorescent group has yellow-green or yellow-brown colonies. For biochemical test, ay ganito yung TSI nila, K over N or K over K. So are alkaline over alkaline or alkaline over neutral, but they are motile, they produce oxidase and do not produce hydrogen sulfide. Fudge typing or phage typing. Fudge typing in short for bacteriophage. Kaya ano ba yung mga bacteriophage? Okay? Ang bacteriophage, sila yung parang bacteria ng mga bacteria. Bacteriophage is a virus. Okay? Kaya sila nasabing bacteria ng mga bacteria. Kasi ang mga viruses mas maliliit naman compared sa mga bacteria. Okay, bacteria of bacteria sa bacteriophage. Okay? Kasi ang bacteriophage, they are viruses sila. Okay? Okay, ang fudge typing or phage typing, so this is a method ang description ng book, this is a phenotypic method that uses your bacteriophages or fudge for short for detecting and identifying the single strain of the bacteria. Okay, so since they are viruses, they can infect bacteria and can lead to lysis of bacteria. So this test allows the identification of bacteria by testing the vulnerability to bacterial viruses that results to lysis. Okay, next is your Acinetobacter, the member of the family Moraxelliaceae. As you've mentioned, this is the second most commonly isolated non-fermentative gram-negative bacillus next to your Pseudomonas. They are obligate, aerobe, and is non-motile and isolated from hospital equipment. Some species may be mistaken as a member of Neisseria species under a microscope, while other species have the tendency to resist alcohol decolorization. They can grow at high temperatures, 43 degrees Celsius. So they are sometimes confused with Neisseria because under the microscope, they also demonstrate Coccobacilli. Okay, di ba yung Neisseria natin, they are Diplobacilli, so parang bacilli na nagtabi. Ito naman, Coccobacilli, they demonstrate this appearance, so para siyang Neisseriae. Ang kaiba lang si Neisseriae, kapag nakita ka ng intracellular in your white blood cells na Neisseria, ay na bacterial cell, you can narrow it down na hindi ito Acinetobacter since it has infected yung white blood cells. So probably this is Neisseria, especially kapag yung specimen mo from your genitalia. Okay, culture, MacAgar, the colonies exhibit a purple color. Parang ganito, biochemical test, they are catalase positive and oxidase negative. Related infections, UTI, pneumonia, endocarditis, meningitis, and cellulitis. Okay, the species of Acinetobacter are Baumannii. Kaya ang basa daw dito, Iwaffi, and Hemolyticus. So your Baumannii and Iwaffi, and Hemolyticus are non-glucose oxidizing compared to your Baumannii, which is glucose oxidizer. Your Stenotrophomonas maltophilia, the third most commonly isolated non-fermentative gram-negative. Okay, sino yung pinakauna? Si Pseudomonas. Okay. Second most commonly isolated si Acinetobacter, and yours truly, yung third most commonly isolated is your Stenotrophomonas maltophilia. They are also strictly aerobic and motile, can grow at high temperatures, and can contaminate blood-drawing equipment and disinfectants. So maltophilia, okay, history ng word. Under the microscope, they are short to medium-sized, gram-negative straight rods. Okay, in blood agar plate, they exhibit lavender green to light purple pigmentation and an odor similar to an ammonia. So, medyo may pagkapanghi. In MAC colonies, they exhibit blue color. Okay, ang biochemical tests are as follows. In antimicrobial tests, used are broth microdilution and E-test. Okay, and related infections, endocarditis, bacteremia, and wound infections. Another is your Burkholderia. They are obligate, aerobe, and motile bipolar flagella except for your Burkholderia maliae. They're generally non-pathogenic. They are also generally an animal infection. They're acquired through contact with heavily contaminated medical devices and grows well on blood, agar plate, chocolate agar plate, in MAC, or your primary mediums. Okay, under the microscope, okay, they are medium-sized, gram-negative straight rods. Okay, biochemical test, they are catalase positive and oxidase negative, and these are the species of the Burkholderia. Cepacia, Pseudomalleae, and Gladioli. Okay, your Burkholderia cepacia, they're isolated in anesthetics, nebulizers, detergents, and disinfectants, and can grow well on most culture media or your primary medias, but may lose its viability on the former in the 3 to 4 days. So BAP and MAC, pag old medium na, hindi na siya viable for testing for your biochemical tests. So in blood agar plate, the colonies exhibit a non-wrinkled yellow or yellow-green color like this one. They are weak positive oxidase reaction and positive in lysine decarboxylase and ONPG. Pneumonia in patients with cystic fibrosis is the related infection. Okay, they are also the agents of glanders or Farcy disease. So they are infection of horses and donkeys. So ganito ang itsura, but they can still, ang tao, they can, people can get the disease, okay, from these animals. Potential bioterrorism agent and only non-motile member of the genus, Burkholderia. They have variable growth on MAC, and in BAP colonies, they are non-pigmented, and oxidase production is variable. Si Pseudomallei naman, agent of Malioidosis or Whitmore's disease, which is a glanders-like disease. Kapag Burkholderia mallei, glanders. Pag Pseudomallei, Malioidosis. Okay, they are motile with a polar tuft of flagella. Okay, so this is an image from electron microscope. Okay, so tuft of flagella, parang ganito siya. Hindi isa. Okay. Isolated from muddy soil and rice paddies. Okay, you can acquire this by inhaling the contaminated debris of direct inoculation through damaged skin or mucous membranes. The presence of bipolar bodies are noted. Parang ganito, di ba? Ang bipolar, ang stain is more prominent sa both end of the bacterial cell. Parang ganito siya. Kaso sa electron microscope kasi ito kaya black and white. So if you culture them in ash-down medium, they demonstrate dry, wrinkled colonies like these ones, and they have a deep pink color. Although they are an animal infection, humans can still acquire them. Next is Cercogenes faecalis. They are obligate aerobic, gram-negative bacillus, and is motile by Peritrichus flagella. So Peritrichus, as described by the book, a flagella that surrounds the cell, the bacterial cell. So para siyang ganito. Okay? So nakapalibot siya. Hindi siya on both ends. Hindi siya sa iisang end, pero nakabalibot. Okay. Ito, iisa. Pero ganito, in reality, makikita yung Peritrichus flagella. Okay, ang nasa book kasi, halos lahat, no? Halos lahat ng sites may flagella. Pero in reality, ganyan sila. A biochemical test, positive in oxidase and grows in 6.5% sodium chloride broth. And in BAP, they are feather edge, non-pigmented, hemolytic, and have fruity odor, which are similar to that of apples or strawberries. Corn tortilla-like, Pseudomonas aeruginosa, fruity odor, Alcaligenes faecalis. Okay, may kasama rin din, no, sa mga odor and characteristic, color, ayan, ng mga bakterya nyo para mas madali kayong mag-narrow down. The Oligelia, okay, are aerobic rod that may appear coccoid in shape, may colonize the distal urethra and cause a serious and active infection. They are small, pure gram-negative bacilli or bacilli. Hi, please remove this. So in blood agar plate, the colonies are small, opaque, and white, and they do not grow in McConkey agar. In performing your biochemical test, they are positive in catalase, oxidase, PAD, or phenylalanine deaminase. They produce gas, and they are non-sugar fermenters. The Oligelia urethralis are non-motile, and Ureolytica is motile. Okay, next is the Chromobacterium violetium, the only species in the genus of Chromobacterium. They are motile with polar flagella, and they are opportunistic pathogen that causes neutrophil deficiency in the immunocompromised patients, and they can grow well on MAC agar demonstrating a violet pigment. So parang ganito siya. Okay, in oxidase test, they are variable. Another is the Salmonella putrescens. which is commonly isolated from water, dairy products, petroleum gas, and other environmental sources. Pati pala gas, no, may bacteria. They are motile, saccharolytic, and strong H2S producer. They are isolated from humans with ocular infections, otitis media, and septicemia. They are gram-negative rods, and in blood agar plate, the colonies are mucoid and greenish. So, parang ganito siya. Biochemical test, oxidase positive, ornithine decarboxylase positive. And this is the summary of the different characteristic after non-fermentative gram-negative bacilli. Okay, the oxidase, motility, croton MAC, croton, and glucose oxidation. Okay, kung maidadagdag kayo, better. Okay, kung gusto nyo iayos in a good fashion para hindi kayo malito kung ano ba yung positive, better pwede nyo pagtabihin ito at ito. Mga ganong style ba para hindi kayo mahirapang mag-differentiate. Okay. Di pa yan. Bali, 3 topics ang i-discuss natin, no? Para last topic na lang next week. Di bali, mabilis lang naman tong mga to. Like your chlamydia family. So, dire-diretso na natin. Huwag na tayo mag-break para minsan ang break. Ay, chlamydia trachomatis, chlamydia sitachi or sitachi, and chlamydia pneumoniae, the related infections, and of course, yung laboratory diagnosis. Hindi pwedeng mawala yon kasi yun yung trabaho natin in the future. Okay. So, chlamydia. So, they are non-motile. They're small and have gram-negative cell wall. They are obligate intracellular ay organisms that require living cells for growth. So, ang basic requirement nila is living cells so they cannot grow on synthetic plates like your MAC, BAP. Okay, they grow usually in McCoy cells, yung mga ganun na agars, na mga mediums, I mean. So they are called energy parasites because they depend on the eukaryotic cells of their host for metabolism, growth, and reproduction, and they replicate by binary fission. So parang ganito. So this is your parent cell. Diba? I think everyone is aware naman or familiar with binary fission. When we say binary fission, so this is an asexual. Diba? Since from one cell lang naman. This is a sexual reproduction by separation of the body, okay, forming two new bodies. Okay, the process of this binary fission wherein the organism duplicates itself, okay, including its genetic material, its DNA. Okay, then they undergo cytokinesis, producing new organism. Okay, so ganun lang ang binary fission. Basta cellular division, a sexual division. Okay, from parent cell, then there is a replication of your DNA, okay, intracellular, sa loob ng cell, for the cytokinesis, and then magse-separate na yung cytoplasm. Okay, cytoplasm divides. Dito na rin yung segregation of DNA, and the cell splits into two new cells, duplicating itself. Okay? Ang species ng chlamydia nyo, si Trachomatis, si Tachyclam, and pneumoniae. Okay, chlamydia has two morphological forms, the reticulate body and elementary bodies. Okay, note that the elementary body is the infectious form, although they are inactive. Kahit inactive sila, they are the infective body compared to reticulate body, which is the replicative and non-infectious form. Okay, ang nangyayari kasi dyan, itong mga elementary bodies natin, okay, kahit metabolically inactive sila, they infect the host cells against, they infect the host cells when ingested. Okay? Kapag yung, yung host cell, okay, na-ingest na yung elementary body, kunwari yung cell natin, na-ingest na yung elementary bodies ng chlamydia, which is the infective stage or the infectious form, okay, pag they will be infected, they will be, nalilito ako. Yung host cell, elementary bodies. When the host cell endocytose yung elementary bodies, which is the infectious but inactive stage of inactive form of the chlamydia, kapag endocytose ng cell, okay, it will evolve into reticulate bodies. Yung reticulate bodies, they will replicate inside the host cell na nag-endocytose ng elementary bodies since they are the replicative form. Okay? Pero in pain, hindi naman to infectious sa cell. Non-infectious siya. Okay? Non-infectious form, reticulate body. Nakuha natin, basta ganun lang yung, parang ganun lang yung process niya. Before siya maging reticulate body, ay elementary body muna siya. Ha? Madali lang i-explain pero nahirapan nung i-deliver. Okay, I hope you got it naman. Dali lang naman. Okay, your reticulate body directs the reproduction of host cells to their own metabolic needs, so they multiply by binary fission, but generally non-infectious. Okay, elementary body, resembles gram-negative bacilli and with a rigid cell wall and infects the host cell by inducing active phagocytosis, specifically endocytosis, or your cell eating. Okay, gets natin? Two components are the major outer membrane protein or the MOMP. Okay. Kung gusto niyo i-clarify, you can butt in and sir, kailangan i-clarify ulit. Okay, Chlamydia trachomatis, one of the major sexually transmitted pathogens, one of the principal causes of pelvic inflammatory disease or PID and ocular trachoma. Okay, si trachoma, as you can see in the picture, ganito ang itsura niya kapag infected ka. Okay, those affected by trachoma also tend to have eye problems, okay, such as cataracts, okay, or forming of dense and dark pigment, dark granules in your eyes. Okay, that eventually lead to visual impairment or worse, blindness. Pwede kayong mabulag. Can travel through the birth canal, where infants can be infected during birth. Okay. Ano ulit yung infection sa mata? Kapag na-infection ng chlamydia or Neisseria, tama? It's the neonatal, ophthalmia neonatorum. Okay, ano ulit yung pinapatak sa mata? It's silver chloride. Okay? Natural host are us, humans. Okay, BioVars, lymphogranuloma venereum or the LGV, mouse and pneumonitis trachoma. So this is an example of lymphogranuloma venereum. This is characterized by, as you can see, a small papule, often asymptomatic skin lesion, followed by, kapag nag-worsen ito, it will affect the lymph nodes of the groin and pelvis, causing lymphadenopathy. And the serovars are the following: serotypes A, B, B, A, and C antigens for endemic trachoma, serotypes L1, L2, L2A for lymphogranuloma venereum, serotypes D to JA, your PID, urethritis, cervitis, epididymitis, and inclusion conjunctivitis. Okay, so related infections and disease, as we've mentioned, trachoma, the inflammation of the conjunctiva that can lead to blindness. Okay, ang TRIC stands for trachoma, inclusion, conjunctivitis. So they cause distortion of the eyelids, mode of transmission, contact with contaminated object, hand-to-hand contact with carriers, and through vectors or flies. So as mentioned here, the intraperitoneal spread may cause peritonitis or perihepatitis or the Fitzhugh-Curtis syndrome. Intraperitoneal or inside your peritoneum, the organs inside the peritoneum or your abdominal organs in your abdomen. Okay? This causes perihepatitis or peritonitis or Fitzhugh-Curtis syndrome. Okay? Perihepatitis. Okay, hepa. Saan ba ang hepa? Sa liver. This Fitzhugh-Curtis syndrome, this is inflammation of the liver capsule without the involvement of its parenchyma. Ano ba yung parenchyma? Ito yung basic functional unit ng liver. So ano ba yung basic functional unit ng liver? Anatomy 101. It's your lobules. Tama? Next is your LGV, okay, or lymphogranuloma venerealium. So as we mentioned, sexually transmitted sila and present a small painless ulcer or papule that appears initially and then nodules develop after several weeks. And inclusion conjunctivitis like this one, yung picture dyan, ay characterized by abundant eye discharge and a swollen conjunctiva that affects infants who acquired it through aspiration or ocular exposure during birth. Okay, pero na-acquire din ito ng ng adults. Okay, sa chlamydia. Chlamydia infection. So laboratory diagnosis, you culture them on the following media: buffalo green monkey kidney cells, HeLa 229 cells, HEPA2 cells, McCoy cells, and cycloheximide treated McCoy cells. Okay, so after 48 to 72 hours of incubation, the monolayers are stained with iodine or immunofluorescent stain to identify the presence of inclusion bodies. For cytologic exams, okay, so basically, ang ginagawa dito is smears. So this smears, yung sa cytologic exams, this is a serological test done for the chlamydia trachomatis on genital smears. Okay, and a direct fluorescent antibody DFA staining method that utilizes the fluorescence isothiocyanate conjugated monoclonal antibodies is used to identify the organisms of infected cell. Okay, so this detects the monoclonal antibodies. Okay, antigen detection and nucleic acid amplification. Nucleic acid amplification is the most sensitive method for detecting C. trachomatis using the specimen and the cervical and urethral swabs. Okay, for serodiagnosis, okay, the lipopolysaccharides or the elementary bodies with deoxyoctonate is the primary antigen that can be identified in the genus-specific test. Okay, so these are IS subjects. So can we leave it like this na lang para, sige na, i-discuss na lang. Kahit touch lang, no? Para kahit paano may background. Yung complement fixation, so this is a test for specific antibodies in patient serum. So for example, yung example dito, ang complement fixation, you have two samples here. So this is from your patient sample and with antibodies siya, okay, siguro na-infecto ng chlamydia. So nag-produce ng antibodies and then ito yung normal patient without infection, so walang antibody production. Ngayon, in complement fixation, ang una mong gagawin dito, i-hit mo tong dalawang to. The purpose of heating is para ma-remove yung antigens para ma-remove yung complements na present dito and even the antigens since they are heat labile, okay? Para ma-remove si antigen or your chlamydia species. So your antibodies will still remain, pero ma-remove yung complement kung present man and antigens. So kunwari nawala na sila. So after heating, okay, ang naiwan na lang dito is yung antibodies, yung mga Y na yan. Wala na tong mga to. At sa normal serum, wala na rin tong complement factors na to. Tapos ang gagawin dyan, after mong ma-hit, magpapasok ka ulit ng so this is a serum, no? Magpapasok ka ulit ng complement and antigen. So parang niloko mo lang yung sarili mo, no? Magkataas mo silang i-hit para mawala, papasukan mo ulit. Pero ang significance nun, yung antigen, magbabind siya sa antibody, forming antigen and antibody complex sa patients na may infection. Ngayon, dito, since walang antibody, mag-i-stay lang yung complement dyan and yung antigens, since wala namang antibody. So, ang next na gagawin, maglalagay ka ngayon ng cell or RBCs sa dalawa. So, dito, since yung antigen nag-bind na si antibody, so, walang maglalise sa cell since yung antigen is na-fix sa antibody, including the complement. Okay? Yung complement factor. Okay? So, yung cell, andyan lang, palutang-lutang, without lysis. Ngayon, dito naman, sa normal serum, Since walang antibody, after mo magpasok ng cell dito, since free-floating lang dyan yung antigen and the complement factors, mag-a-attach sila sa cell and will lead to lysis of the cell. So, ang positive dito, walang lysis. That's the complement fixation. Absence of lysis, positive. Negative lysis, ay pag walang lysis, negative. Kapag positive ka sa infection, walang lysis. Okay? Since andito yung antibodies. Wow. Diba maaral nyo pa naman yan sa IS? The complement fixation identifies family reactive antibodies, the use for diagnosis of your LGV, and genus-specific antigens can be detected by using this method. So positive result, titer equal to 1:64 titer. Another is your microimmunofluorescence test or your MIFAC. Ang microimmunofluorescence test, this is an indirect fluorescent antibody technique that enables yung observation of the binding of the antigen to the antibody that is detected by fluoresceins or fluorescein conjugated antiglobulin to the corresponding antibody molecules. Yeah, leave it like this muna. Kung ano yung nandito, yun na lang muna since bacteriology naman ang topic. But I'm not discouraging you to read books, no? That can further let you understand yung mga ganitong terms. But soon you will know yung mga ito na din. Okay, preferred method for identification of antibodies to Chlamydia trachomatis and can be used for the diagnosis of LGV trachoma, inclusion conjunctivitis, and Chlamydia neonatal infection. So positive result, IgM titer of 1:32, and your immunoassays, your EA that detects either the outer membrane lipopolysaccharide chlamydial antigen or the MOMP antigen. Next, your Chlamydia psittaci, a causative agent of psittacosis or ornithosis that is commonly found in bird species such as the parrot. So, ayan, parakeets, chicken, and ducks. So, mode of acquisition, inhalation of infected aerosols from dried bird excreta or handling of infected birds. Kwan lang, di ba kwan na may bird flu na sa Cagayan? May ano na silang nag-positive. So, yeah, ingat-ingat. So, psittacosis, this is an infectious disease usually spread to humans by infected birds, specifically mga parrot families. So, kapag infected ka na ganito, siyempre, infection, magkakaroon ka ng fever, chills, cough, sometimes breathing difficulty din or pneumonia. Kapag hindi ito na-treat, no, the disease can be, siyempre, walang medical intervention, pwede yung lumala. At kapag lumala ng lumala, lead to death pa rin. Okay. Specifically, sa mga matatanda, sa mga lola at lolo natin. And even in sa mga bata. Kasi ang mga bata at matatanda, they are the population. I mean, yung mga neonates, hindi yung mga teens. Kasi active yung mga antibody production ng mga neonates and old people, they are prone to infection since yung immune system nila, as we age, pababa ng pababa. Okay, but in 30s and 40s, okay pa yan. So, sensitive method, direct microimmunofluorescence, and precautionary measures only laboratories with biosafety level 3 facilities can perform the isolation and cultivation of this specimen. Chlamydia pneumoniae, formerly known as Chlamydia pneumoniae strain TWAR, or the TWAR strain, a human pathogen that is transmitted through aerosol droplets and one of the major causes of infectious respiratory disease. Preferred method, microimmunofluorescent assay, and the same culture media sa mga living cells. Init. Next. Ayun. Okay, so this is a differentiation of Chlamydia species. Chlamydia trachomatis, psittaci, pneumoniae. Wait lang, init. Okay, susceptibility sa sulfonamides, the only resistant species is trachomatis. Susceptible, sorry, and ang resistant is psittaci and pneumoniae. Okay, ano bang ino-note dito? Basta i-note nyo na lang yung mga kakaiba. Madali lang naman silang tandaan. Okay, kung glycogen-containing inclusions, si Trachomatis. Okay? And last, your cell wall deficient bacteria, which is your Mycoplasma and Urea plasma. Okay. Mabilis lang to, mga 7 slides or 8 slides lang. So, Mycoplasma and Urea plasma are the molecules. They are considered as the smallest free-living organisms. Since absent ang cell wall nila, ano ang counterpart? Yung sterols or the cholesterol sterol for membrane function and growth. Ang species, si Mycoplasma pneumoniae, hominis, fermentans, spirum, penetrans, and Ureaplasma urealyticum. So in Mycoplasma pneumoniae, okay, si Mycoplasma hominis, hindi to bominis ha. Hominis, the colonies exhibit fried egg appearance, so parang ganyan, unaplated medium, and the Ureaplasma urealyticum exhibit dark brownish lumps on A7 or A8 agar medium. So ganito sila. So they can be recovered from the genital tract of healthy adults and can cause prostatitis and pelvic inflammatory disease. So for laboratory diagnosis, there is no direct method or gram staining can be used for identification. Right. You culture them using beef or soybean protein with serum, fresh yeast extract, biphasic SP for medium, and the following. Okay, swab samples, swab for sample collection, calcium alginate integrin swab. Okay, gawa rin kayo no ng sa reviewer nyo ng mnemonics kung sino yung mga species that are strictly collected using calcium alginate and Dacron swabs. Andyan si Streptococcus pneumoniae, si Mycoplasma pneumoniae, Neisseria gonorrhoeae, di ba? Calcium alginate. You make one para hindi nyo nahahanap-hanapin pa kung saan. Okay, transport medium, SP4, sucrose phosphate buffer, horse serum, and neutral red. Okay, you culture, in culturing them, may mga specificities sila, which is, I think this is self-explanatory naman na. Okay? Mycoplasma hominis, capable of growing in BAP and CAP. I no need to explain. Blood culture not recommended for Mycoplasma and overlying suspicious colonies of 0.5% guinea pig and RBCs in phosphate buffer saline is the definitive identification of Mycoplasma pneumoniae. Serodiagnosis using your ELISA and manganese chloride urea test, the rapid identification of Ureaplasma urealyticum. Yan. Ano pa ba? So, sa dito, yung Ureaplasma urealyticum, ang positive for urease test, okay, they indicate brown halo surrounding the colonies. If you stick it, tapos may brown halo sila, oh, they are Ureaplasma urealyticum. Brown halo. Okay, and this is the comparison between the common bacteria and atypical bacteria. Okay. Yeah. Rickettsia, this is Rickettsia pa pala, no? So may 2 topics pa tayo next meeting. Rickettsia and your miscellaneous bacteria. Okay, so who are obligate intracellular, yung bacteria na Ureaplasma. Ano to, ba't bacteria? Obligate intracellular, Rickettsia, Chlamydia, Mycoplasma, DNA, RNA, Rickettsia, Chlamydia, and Mycoplasma. A presence of ribosomes, peptidoglycan in cell wall. Since walang cell wall si Mycoplasma, wala silang peptidoglycan. Replication by binary fission. Lahat sila. Okay, these bacteria, other than Rickettsia, Chlamydia, and Mycoplasma. Okay? Bacteria other than Rickettsia and Chlamydia. Okay, kasi as you can see here naman, since may peptidoglycan, lahat naman ng bacteria may cell wall, di ba? Sila Mycoplasma, Ureaplasma lang naman ang wala. Okay, and ito na ba ang last? Okay, that's the last slide. Yes, last meeting na next week. So, any questions so far? Meron? Dismiss na. So, since mahaba kasi yung quiz nyo next meeting, Enterobacteriaceae and your gram, ano yung isa? Non-enteric GI pathogens. So, please use the remaining time in scanning your notes or pag hindi nyo pa na browse yung lecture video, please have time to browse those videos. No, mahaba-haba yun. Sa Enterobacteriaceae, you will have a 30-point quiz and non-GI pathogens, I think 10 items. Okay, yon. So next meeting, pakisabi sa instructor nyo before our subject that you have to be on your GMAT in time. So hanggat maaari sana wag na silang mag-request or mag-favor sa inyo ng extension of time kasi naghahabol din tayo. Next meeting, as you have seen in the announcement, my quiz, then after yung quiz, i-discuss natin yung last topic, your miscellaneous bacteria, the rickettsia, and the miscellaneous bacteria. Then after yung discussion, yung reporting, yung sinasabi ko kanina, which is for applied bacteriology. Huwag kayong mag-alala kasi yung reporting nyo, by group naman yan, pwede nyong, sa kabilang section kasi, tinanong nila kung pwedeng recorded lecture. As long as nakikita kayo dun sa, yung parang ginagawa ko ding recorded lecture, yung meron kayong nakikita para habang nakikinig kami, nakikita namin yung nagsasalita. Okay? Para mas, hindi ko alam. Kasi ako mas naiintindihan ko yung topic pag nakikita ko yung nagsasalita. Okay? So, can I request everyone to open up their cameras to see kung may nakikinig ba sa akin or dadalang ako ng dada dito. Okay? And we'll have our photo ops for your class participation. Class participation para hindi naman na tayo nagpaparecite, sayang din yung idle time at naghahabol tayo ng lessons. Gamitin niyo yung remaining time for review para Desiree, are you here? Ayun, meron siya. How about si Alisa? Si Marigold. Are you here, Marigold? Si Paula Marie. Si Rose Connie. Paki-open na lang yung cam. Ay, meron siya. Faren Kalingayan. Blanco.
Sir,
meron siya. Okay, Blanco Venson. Are you here? And Katrina Marana. Ba't tayo niyo mag-open cam? Juan, ba kaya ba yung batch na na first year, hindi pa kayo nag-meet personally? Ito ba yun? Ay ba? Ay, kayo na. Open nyo na yung camera nyo para magkitaan naman tayo ng mukha. Ayaw nyo. Ayan. So, next meeting, yung mga announcement, and magsalita kayo lahat sa reporting para naman marinig ko din yung mga magagandang boses nyo. Now, so, okay, wala nang questions. 1, 2, 3, smile. This is for your class participation. Sa mga ayaw mag-open, ayan, do this na lang para alam ko nandito kayo. Okay, 1, 2, 3, smile. Okay, ayan. Okay, use your time to review sa long quiz nyo next meeting. Okay, and prepare for your report na rin. Okay, bye-bye. See you next week.